So as Mark said, I am Niranj Chandrasekaran. I am a postdoctoral associate in the imaging platform at the Broad Institute. Today, I'm going to be talking to you about the updates from the JUMP Cell Painting Consortium. Many of you are probably aware of this consortium, but for those who aren't, I will give you an introduction about this consortium and its efforts. And for the first 10 minutes or so, I will be talking to you about some of the experimental updates that have happened in the consortium over the last year and a half. And after that, Paula will be talking to you about the computational updates. At the bottom of the slide, I have provided a link to this very slide deck. So in case anyone wants to follow along, please feel free to do so. All right, so what is the JUMP Cell Painting Consortium? Well, JUMP stands for Joint Undertaking for Morphological Profiling, and we'll be using cell painting as the assay for performing morphological profiling. The main goal of this data set is to generate the world's largest publicly available cell painting data set. So this effort will involve the creation of over 30 million images displaying cells that respond to over 140,000 chemical and genetic perturbations in 5 replicates. And the most interesting thing about this experiment is that each of these 5 replicates will be generated. The data for that will be generated in 5 different sites. That makes this data set extremely valuable because of the diversity, the different kinds of equipments and personnel that went into generating this data set. The other goals of this consortium itself is to create a community of people working in morphological profiling using cell painting, and also to develop best practices and communicate it to the rest of the community. So at the moment, there are 10 for-profit partners who are part of this consortium and 2 non-profit partners. We also have a bunch of collaborators whose contribution, the scope of their contribution range all the way from administrative assistance to even data production. So if there are people in this in the audience who are interested in collaborating with us after I give the talk, please feel free to reach out to us. I've also provided the link to this to the website where you can find a lot more information about this project. So the project was originally funded in July of 2019, and the public release of the data that will be generated in this project will happen in 2022, November of 2022. So far, we have been primarily working on experiment planning and optimizing experimental conditions using a number of pilot experiments. And we've also, as I said, the replicates for this experiment will be, the experiments on these replicates will be performed at different sites. So we have also been involved in extensive component exchange between the different partners in the consortium. Currently, we are working on developing computational methods for analysis, and also data production is ongoing. Once the data production finishes around the end of this year, we will embark on data analysis. So for those of you who might be new to the field of morphological profiling itself, and specifically in the context of cell painting, I wanted to show you this schematic, which summarizes the entire experiment. So what we do in this experiment is take cells. It could be cells from healthy or diseased patient cell lines. And they could be subjected to perturbations like chemical and genetic perturbation. And then using a high throughput assay, we stain the cells and then image them as well. And an example of a high throughput assay, and the one that we will be using for this data set, is cell painting. So in cell painting, we have there are 8 cellular components that are stained using 6 different fluorescent dyes, and then they are imaged in 5 different channels. So once these cells that have been stained are imaged, then features are extracted from the cells in this step. And this feature extraction can happen either using a classical approach with Cell Profiler, or using a more modern approach using deep neural networks. Once the features are extracted, they are assembled together to form these profiles. You could have profiles at a single cell level, or you could have a well-level aggregated profile. These profiles are what we call the morphological profiles. And once you have these profiles, then you could do a number of different downstream analysis to extract valuable information from these profiles. So yeah, so that's a summary of a morphological profile experiment using cell painting. And I will now talk to you about a couple of, give you updates on a couple of things that we have been working on in this project for the last year or so. So the two outcomes that I want to discuss are first, optimization of a number of experimental conditions in this morphological profiling pipeline. And the second is about a pilot data release, a data set that I believe many of you in the audience may be very interested in exploring. So first, looking at the optimized experimental parameters, if you want to run cell painting experiments, these are the parameters that you would want to use while running experiments. The results of this are being written up currently and will soon be submitted. So let's come back to this schematic showing you the entire morphological profiling pipeline. And if we just focus on the left half of this pipeline, where it involves all the experimental steps, there are a number of parameters that one could optimize. Some of these have to do with the type of cells and all the associated parameters with cells. Others are associated with stains. And then some involve microscope settings and fields of view. So there are many parameters that could be optimized, but these are the ones that we decided to focus on for this particular project. So we performed a number of experiments and under different conditions, and we now had to compare the different conditions to see which is the more optimal value for certain parameter. And the way we did that was by using a bunch of metrics, a class of metrics that we call the percent scores. And these values are dependent on calculating distributions of median pairwise correlations. And depending on the metric, the pairs actually vary. So for the metric percent replicating, the replicates form the perturbation pairs, while for percent matching, it's perturbations that belong to the same class. So what these metrics basically measure is how many perturbations correlate strongly with their replicates. That's what is given by percent replicating. And percent matching tells you how many perturbations correlate strongly with other perturbations that belong to the same class. So the maximum value that both these metrics can have is 100%. And the closer the value to 100, the better. So that's the most important thing that you need to get out of this slide. So this is a sample experiment. This is an example result that I wanted to show you from this particular experiment. Here, what we did was we compared a number of different parameters that I mentioned previously in one of the slides. We have two different reagent vendors, Thermo Fisher and PerkinElmer. We have two different scope settings, that is either using wide field images or capturing wide field images or capturing confocal images. And we have three different reagent concentrations here, just written as condition A, B, and C. So here we plot percent replicating versus percent matching. And what we see here is that when you look at this plot, you can already come to a few conclusions based on just this simple plot here. So you can see that between Thermo Fisher and PerkinElmer, you can see that the PerkinElmer values are slightly higher in both the axes, which is a good thing. So maybe PerkinElmer is performing slightly better. When we compare the scope settings, we find that the performance is about qualitatively very similar across the two conditions. So maybe there is no clear winner. And when it comes to the dye concentrations, we find that condition A and C seem to be separated from condition B. So they seem to perform better than condition B. So these are the kind of conclusions that we come up with based on these two metrics. And we performed experiments to test all the conditions that I mentioned previously. So here's a summary. There is a lot of information here, so probably you may not be able to focus on every one of these. But what I want to mention here is that the non-grayed out text here are the optimal values that we found out from our pilot experiments. And these are the values that we will be using for the main data production phase of this project. For For example, the reagent vendor, we decided to go with PerkinElmer over Thermo Fisher, and for cell type, we went with U2OS over A549. Many of these decisions were made solely based on the metrics that I defined previously, the 2% scores. But sometimes we also made decisions based on which one was cheaper, both time-wise and cost-wise. Because some of the times, both conditions perform very similarly, at least based on the metrics. Let me quickly move on to the other topic that I wanted to talk to you about, which was the pilot data set that we recently released. So this data set, we call it the CpJump1 pilot data set, and it's a well-curated, annotated data set. It consists of 3 different plates: a compound plate, a CRISPR plate, and an ORF plate. And compound, of course, is the chemical perturbation plate, and the CRISPR and ORF are the genetic perturbation plates. And we have a total of 302 compounds in this experiment and 160 genetic perturbations. We have 2 different cell types, U2OS and A549, and we also have different time points, short and long, for both genetic perturbation and chemical perturbations. What is the most interesting thing about this data set is that each gene in this experiment is targeted by at least 2 compounds, 2 CRISPR guides, and 1 ORF. Therefore, we have multiple different modalities of perturbations for the same gene. Therefore, we may be able to perform experiments to match across modalities, which is the exciting part. So there are a number of potential uses for this data set. I'll be here showing you 2 use cases that we tested, but of course, there could be many other things that one could do with this particular data set. The first one is benchmarking representation learning methods. So here what we did was we took the features that we extracted using Cell Profiler, shown here in blue, and also using Deep Profiler, which is a counterpart of Cell Profiler, but uses deep learning to extract features. And here we used a pre-trained network, efficient net, in order to extract the features from the cells. So what you can see here in general is across the different time points and across the different cell lines, the chemical perturbation, the compound one, seems to have a higher percent replicating compared to the ORFs and CRISPRs, the genetic perturbations. And that seems to be kind of consistent for both Cell Profiler and Deep Profiler. So one could develop their own representation learning method, and they could use this particular data set to benchmark their methods. The other application, or the other use that this data set is much more suited for is benchmarking gene compound matching methods. So as I already said, we have perturbations, both chemical and genetic perturbation for the same genes. So we could get profiles from compounds and also from CRISPRs and ORFs, and we could try matching. So using a very naive approach based on percent matching, we found that about 7 to 11% of the genes, the compounds and the genetic perturbations show a very strong correlation. It's possible that with a much more complex method, much more advanced method, one might be able to increase this performance significantly. A little bit more about this data set. There are 2.73 million images in 5 different fluorescent channels. And there are 3 bright field Z-planes. And we have provided Cell Profiler derived features for all these images, but we also have provided Deep Profiler derived features for a subset of them. And one could download this data like today from this link here. So yeah, those are my slides. I want to thank all the 100+ scientists in this consortium who have contributed to this project. And I especially want to thank the people I have listed on this slide who were important, who helped me generate this particular data set that I shared with you today and to analyze them. And I also would like to thank Massachusetts Life Sciences Center, the funding agency. Without their funding, this project would not have been possible. So over to Paula now.